Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Hisham. I'm Director, Institute of Bioproducts Development. And I'm honored to welcome you today for our regular seminars for biotechnology, science, business, and society. And today is webinar number 36. For today, we have a very special guest, our friend, Dr. Ahmed Mediani, who received his bachelor degree in agriculture and nutrition from Algeria. And he obtained his master and PhD degree from food science from University Putra, Malaysia. And he's currently serving as university lecturer in Institute of Systems Biology in Piosis, which is University Kebangsaan, Malaysia, UKM. He has published a number of research papers. As I know him personally, he published almost 100 publications and peer-reviewed international journal in a very short time. And he's considered one of the very active researchers in national level in terms of herbal technology. And as a young and dynamic, and we do a lot of work in many areas related to food science, metabolomics, and food chemistry. He's talked with us today about timely topic, which is very of high interest for everyone, I think, which is metabolomics as the holistic approach in natural products. With this, I would like to say thank you very much, Dr. Ahmed, for accepting our invitation to be our guest speaker for today. Thank you very much.
Thank you very much, Prof. Hisham, for the kind invitation. I am very pleased and honored to be with you today. Can I share my slide? Please allow me to share my slide. Can you see my slide?
Yes, Dr. Ahmed, I can see your slide. Thank you.
Okay. Bismillahirrahmanirrahim. Wassalatu wassalamu ala sayyidina Muhammad wa ala alihi wa sahbihi wa sallama taslima. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Again, thank you very much, Prof. Hisham, for the kind introduction. I really appreciate it. It was very nice and very kind of you. Again, thank you for the invitation for this webinar series. I am Ahmed Mediani. Today, I want to present to you about metabolomics as a holistic approach in natural products. I am from Symbiosis UKM, as a Prof. Hisham mentioned just now. This is my email for anything you want to contact me. Okay, before I start, I want to introduce you to my institute, Symbiosis UKM. Could you please play the video?
DNA, suatu istilah yang sentiasa dikaitkan dengan kes siasatan forensik dan ia merupakan antara elemen utama dalam penyelidikan biologi moden. Tetapi pernahkah anda terfikir DNA ini adalah merupakan sebahagian daripada data biologi yang memainkan peranan penting dalam perawatan penyakit tumbuh-tumbuhan dan boleh membantu mengatasi masalah ekonomi negara? Sehubungan itu, sebuah institut penyelidikan telah diwujudkan di UKM untuk mengkaji dan memanfaatkan bidang yang menghasilkan lambakan data biologi. Ditubuhkan pada tahun 2005, Institut Biologi Sistem yang lebih dikenali dengan Symbiosis mengorak langkah mendepani bidang bioteknologi moden di negara ini dengan wawasan utama untuk memantap dan memacu penyelidikan biologi sistem dan biologi sintetik. Symbiosis memacu kajian terkehadapan melalui 3 pusat kecemerlangan dengan menggunakan teknik terkini dalam bidang biologi, sains komputer, teknologi maklumat dan sains data. Imbiosis membawa era baru dalam kajian produk semula jadi negara dengan mengaplikasi teknologi omik dan biologi pengkomputeran untuk memantapkan bidang phytomyceutical dan data raya biologi dalam mengurai pelbagai misteri sifat biomolekul yang berpotensi dalam pelbagai industri. Imbiosis juga mempelopori bidang biologi sintetik dan penyuntingan genom di negara ini ke arah penyelidikan translasi dan pembangunan bioproduk. Untuk memacu inovasi ke tahap yang lebih tinggi, Imbiosis turut bekerjasama dengan industri dalam dan luar negara.
We have been working closely with Imbiosis since 2015, where we conducted fungal metabolomics.
We have developed a species metabolite relational database called NAPSA, and also we have developed several novel algorithms for extensive analysis of key biological data,
where we exchange expertise in natural products, phytopharmaceuticals, and metabolomic studies.
In this collaboration, we have identified fungi that are able to live solely based on plant lipids.
As one of the first systems biology institutes, I believe that Imbiosis provides a well-structured and strong platform in omics research, especially for young scientists.
Berbekalkan budaya kecemerlangan dan prospek kejaya, Imbiosis menawarkan program siswazah bertaraf antarabangsa untuk menghasilkan saintis biologi alaf baru.
Di sini, kami diberi pendedahan dan latihan dalam biologi sistem dan biologi sintetik.
Saya memilih Imbiosis kerana institut ini mempelopori kajian inovatif yang berteknologi tinggi dalam bidang omics di Asia.
Imbiosis adalah satu-satunya institusi di Malaysia yang mempunyai kapasiti pemprosesan data biologi yang tinggi.
Saya telah didedahkan ke pelbagai teknik makmal yang membolehkan saya menjalankan kajian untuk menjawab soalan-soalan penyelidikan.
Saya dapati Imbiosis telah mendirikan persekitaran pembelajaran dan penyelidikan yang kondusif.
Imbiosis memberikan peluang untuk kami menjalankan penyelidikan rentas bidang, terutama di dalam bidang bioteknologi dan farmaceutical bagi penghasilan bioproduk.
Sertailah Imbiosis untuk sama-sama mengilham harapan, mencipta masa depan demi mencapai visi dan misi UKM.
I hope this video provides you a summary about the research done in Imbiosis. You are welcome for collaboration. We have experts in all the omics approaches, including proteomics, genomics, and metabolomics, and other omics, and bioinformatics also. I am also proud that I am alumni UTM. I came for the workshop last time in July 2022, and now I am alumni from there. Here I am with Prof. Hisham, who gave me the certificate. Thank you very much. Let me start my presentation. As all of you know, that the traditional medicine or the traditional natural products, especially plant, as the source of the drugs. They are mainly, since the ancient time, they are the main source of the drugs. And most of the medicine, they are based from these plants, especially the secondary metabolite, which been successful source for potential drug use. And it is especially in China or Malaysia, they are really based on this secondary metabolite. And most of the research and its origin in ethnopharmacological practice, then they need to do the safety and others. I will show you in the next slide how Metabolomics can do this, the safety analysis and other efficiency and other things. And modern natural product research involve a range of approaches, especially for the people, the one they are from natural product, they know the isolation is one of the approaches that they are doing when they isolate the compound and then test it for biological activity. But we have a problem with isolation. When you isolate the compound and you test it, sometimes you will not find it as a bioactive compound because for the synergistic effect. That's why other approaches as metabolomics can be done because they can study the synergistic effect. And Malaysia is one of the biodiversity country. It has more than 50,000 species from plant and other natural product resources. That's why we need to make use of these resources. And from the drug, it can be from natural product, any plant or the marine. We need to do this characteristic. We need to study the safety, especially sometimes if the drug is efficacious and the quality and the consistency is okay, but it is toxic, it's no point. That's why we need to make sure the drug is safe. And the efficacy, of course, we need to be efficacious. If it is not, if efficiency is not high, then it cannot be go for the stage to the drug. The quality, what I mean by the quality, sometimes, especially the people doing isolation and other approaches, when they come with the batch, because they sometimes when we do the isolation, we go for the batch of 100 kg. Sometimes when you have another drug, another batch, the bioactivity, when we test it, it will be totally different. Sometimes in the first time it will be active, but we come with another batch, it will not be active. That is the quality. That's why we need to study the quality and you need to be consistent. When we bring the batch from one supplier, better to keep getting the sample from the same supplier, and that supplier needs to control many factors that can affect the compound or the sample for not degraded. That's why the method is required, should be faster and efficient. Metabolomics is one of the approaches that can be used to study all these characteristics. I came to the definition of the metabolomics. Most of you, I think, know what is the metabolomics. Metabolomics is a downstream product of the gene expression, and it can reflect the functional level of the cell. And metabolomics, it is qualitative and quantitative. What I mean by quantitative, it can qualify all the compound or can quantify. When we do the sampling, we do the analysis, because metabolomics is when we couple one analytical tools, which is SPLC, LCMS, GCMS, or NMR. Sometimes we use FTIR, but FTIR cannot be, most of the people nowadays, they are not using because it can only show the functional group. Then coupled to the multivariate data analysis. Then we do quantitative and qualitative. In terms of qualitative, we know all the compound found in that sample. And quantitative, sometimes when we compare two different species, they have the same compound, but in terms of the quantity of the amount of the sample, it will be different. Then it can do the quantitative analysis. As compare metabolomics to other omics, we can see that metabolomics is a downstream. Whatever changes during the functional level of the cell, it can appear in the metabolite. That's why sometimes if we eat something, like if we eat the durian, if we go to check the gene, we will not see maybe the one who take the durian and the other one who doesn't take the durian. We want to differentiate between them. We cannot see it in terms of the gene or the protein. But if we analyze the metabolite, you can see definitely the changes in the urine or in the blood or in the sample from when we do the metabolite. And this is the general outlines of the metabolomics. We start for the sampling. Sampling, we need to have replicate. For metabolomics, especially because we are doing multivariate data analysis, the statistical analysis need replicate. What I mean by replicate, we need the biological replicate. Every one sample is one biological replicate. Biological replicate is very important. Then sample preparation. Sample preparation, we need to make sure that the sample preparation that you are doing should be very fast and it cannot degrade the metabolite, or at least it can cover most of the metabolites of interest and make it as simple as possible. Because when you put many steps, then it can degrade most of the metabolite. That's why we need to make it fast. Data acquisition, as I mentioned just now, we can do it based on HPLC, LCMS, GCMS, or ANAMA, FTAR also. And the QC sample is very important for the MS analysis. Later on, I will show what is the QC sample. The data pre-processing and data treatment is very important because we need to have a good sample and a good data before we proceed with the multivariate data analysis, which is the statistical analysis. It can be supervised and unsupervised method. I will show you what is the difference between supervised and unsupervised. Metabolite identification. Okay, between the multivariate data analysis and the metabolite identification can be switched. If we do the targeted analysis, maybe we can start with the metabolite identification because we know what are the metabolites of interest. We identify all of them, then we go for the multivariate data analysis. But if you are doing untargeted analysis, better to start with the multivariate data analysis. Then you find what are the metabolites significant. Then you identify them. Because sometimes if you start with the metabolite identification, you waste your time. You identify, let's see, you identify 200 metabolites. After you go for the multivariate data analysis, you will find all the metabolites that you identify, they are not significant. Then it's like you are wasting your time. Better to start with multivariate data analysis first. Okay, for the metabolomic study, especially the people doing plant sample, need to control many factors. The one they can cause some effect to the metabolite and also can cause some differences between the sample before you start, especially the growth condition. Maybe sometimes if your sample is from different farm, the soil can be different, fertilizer, the climate, the watering system can also affect the metabolite. And sometimes some people, they are using the pesticide or insecticide. This product can affect the metabolite. Also, when you have the sample from different location, we know that if you bring the sample, I have one study, I have the sample from Terengganu or the sample from KL. When we compare, we can see that they are totally different. And some people, they have sample from different country. Harvesting time. Harvesting time also can affect the metabolite. When you collect the sample in the morning or in the afternoon, can have totally different metabolite. And also some metabolite can be only find in the morning when you collect the sample in the morning. That's why you need to be consistent. When you are doing metabolomics, you need to collect your sample at the same time. If you collect it in the morning, you need to be consistent with all the study until the end. You need to collect your sample only in the morning. The plant part. When you compare between the fruit, the leaf, the stem, or the roots, or also the seed, sometimes can have totally different metabolite. And most of the people, if you don't have enough sample, maybe you can take the aerial part, just remove this the roots, then you can take the aerial part, whatever above the ground, you can take it and you make it as your sample. For the processing, drying and extraction condition, if you dry your sample using a different drying method, can affect the metabolite. And also the extraction condition, like the oven drying, the extraction when you are using the soaking or the soaking or sonication, can have SFE or different other method can affect. That's why you need to be consistent and take, or sometimes you can compare between different extraction method and choose which one is the best. Different solvent also, because when you have different solvent can also affect the metabolite, especially when you are dealing with the polar or non-polar. If you are going for targeted analysis, you need to make sure that solvent that you are using can extract the compound of interest. Later on, I will show you what is the difference between the polar and the non-polar solvent. And the solution, if you want to control most of the factor, is to plant your own sample. For me and for my students, I always ask them to plant the sample and try at least to control most of the factors that can affect the metabolite degradation or the differences between the samples, especially when you are comparing different species or you have different sample from different place. For the animal, the people who is doing the animal study, also, let's see, if you are doing the rats, the study of the rats, the stress can cause differences in the metabolite. When you have one rat in one cage, can that social interaction, because the rats doesn't have social interaction. That's why most of the time for the animal ethic, they will ask you to put at least 2 rats in one cage. Then they can have social interaction and then they will not have stress. The environment and the condition, the humidity, you need to control the humidity and the temperature. For the animal type, species, and the gender, we can see here in this study, they are doing the obesity. Between the male and the female, they are the same age, but the female gain more weight as compared to the male at the same age. They are taking the same food, at the same time, they are having totally different weight. That's why you need to be very careful. And the housing condition, as I mentioned just now, handling the method. The handling method, for the animal study, it's better for the same person to handle, or at least minimize the people who can enter the room of the animals. Don't put many people to enter the room because they will be stressed. The tissue to be analyzed, this one I will mention, what is you want to focus on the urine or the biofluid or the tissue, depend on your objective, what you want to study. The collection and the extraction condition as the same as the plant sample just now. The solution, you need to have your animal from the same supplier, or you can breed your own animal. As for the protocol, you need to have, for the sample to avoid the degradation, you need to harvest the sample and directly quench with the liquid nitrogen, or you can put it in the -80 to stop the enzymatic reaction for any sample, for the plant sample or for the biofluid. To avoid the degradation of the metabolites, better to quench with the liquid nitrogen or put in the -80. The rest is like the same, just the design just now I show. And for the biofluid, if you want to compare between the blood and the urine, mostly the urine will have most of the metabolite as compared to the blood, but depend on your objective, what you want to see. For the people who are doing the long-term study, then maybe can go for the urine as compared to the blood. And it's based on your design and your objective. You can see the protocol that you are doing, and you make sure that it can be good for your study. As I mentioned just now, the blood, this is the sample on the right is the urine sample. We can see more metabolite from the urine as compared to the blood from the same animal. If you can do it complementary, maybe can have some metabolite can be seen in the serum as compared to the urine, and the opposite. it's better to do both of the sample. And most of the publication now, they are asking to do it complementary. Also for the, I want to focus on the NMR. When you have different frequency of the NMR can show you totally different metabolite. Or also the frequency can see that some of the metabolite with 950 megahertz can be seen very clear as compared to other 500. Or for metabolomics, the minimum one is 500. At least you need to have the NMR of frequency of 500, then you can deal with the metabolomics. But if you have the 1200 now, the latest one is 1200. That one is very good for the, especially for the 2D data. For the NMR study, when you do the analysis, you need to have the buffer to avoid the shifting of the metabolite. When you put the buffer, you can see here the sample on the top that has the buffer with the specific pH. For the urine, we have the pH of 7.4 or the serum. But for the plant sample, mostly we are using the pH of 6. We can see that the one with the buffer, we don't notice any over shifting of the peaks. But when we don't have, in the bottom one, you can see that some of the sample has the peaks, they are shifting. That's why it's very necessary to do the buffer with specific pH. This is also for the water pre-saturation or water separation. When you run the NMR, you need to do the water separation because most of the sample, because you are dealing with totally different sample, you cannot handle the sample or you avoid the water because the sample can absorb the water anytime. You need to do the water separation. And the best protocol is to do the freeze drying. Sometimes you need to do the freeze drying. And also when you run the NMR, you do the water pre-saturation. You can see here, the one without water, the water can suppress other compound or other signals. When you do the water separation, you can see that other signal can be increased. And for the serum or the plasma, we need to do the CPMG. CPMG, this one to reduce the big molecules. Extra step to do the pre-saturation, the water pre-saturation, and also you go for CPMG to suppress the big molecules because the big molecules can interact with most of the metabolite in the serum or in the plasma or the cell. As I mentioned just now, for the multivariate data analysis, we need statistic. And also this statistic need replicate. And they say we need biological replicate. Every one animal or one plant is one biological replicate. But you need to know that the biological replicate can have most of the variation because as compared to the technical replicate, because the technical replicate is the same sample, you process it separately. Then we don't need for the NMR, we don't need the technical replicate. Only biological replicate is enough. But if you want to check your method or your technique, you can include the technical replicate and you can check how your technique is robust or accurate. This is I just want to show what is the sample preparation when you have 3 different biological replicate. You can sometimes you can experience that some of the plant, they will have totally different height. Maybe some of them, they are flowering, the other one is not flowering. They have high variability as compared to the technical replicate. And the technical replicate from the same plant, you take total different sample and you process it separately. But when you process the sample separately and you run from the same sample, you run it different time, that one is not replicate. That's why you need to avoid that. That one is called determination, is not replicate anymore. That's why you need to be careful with that. But for the MS analysis, we can combine between the biological replicate and the technical replicate at the same time. For the animal, every one animal is one biological replicate. But sometimes, if we don't have enough sample, maybe we can combine, maybe 2 or 3 we can consider them as one biological replicate. But mostly for the metabolomics for the animal, we have enough. But if you are dealing with the mice or small animals, sometimes the sample will not be enough to do the metabolomics. That's why maybe you can combine 4 to 5 animal as one biological replicate. And it's better to have the same for the treatment and for the control groups to have the same replicate. For the MS, especially for publication now, they are asking at least 4 biological replicate. But for the NMR, from 6 to 10, at least 6 biological replicates. This is only to show for the MS how to do the biological replicate and the technical replicate. And you measure, when you have the biological replicate, you can see that they are totally different. And then maybe you can combine between the biological and the technical replicate to see your technique. And also, as I mentioned just now, from 4 to 6 replicates, it is okay for the metabolomic study. You can combine both. And from the metabolomics, we have a big data. It is considered as a big data. And you can experience that we have 100 sample from different groups. And this one, every sample has at least 200 signal or 200 metabolite. This thing you cannot handle it by the eyes, or you cannot handle it by hand just to see the differences. That's why we need very powerful statistical analysis, which is multivariate data analysis. And this multivariate data analysis, it depends on which model you want to use, is supervised or unsupervised. Let's see here, the model is perfect, but the data that you process it is not processed properly. That means you are introducing rubbish data to the model. Then you expect that your result will be rubbish. And for the second example, the data, it is processed nicely and the data is good and also the design is good, but the model that you choose for the multivariate data analysis is not good. It is rubbish, then you can expect that your result will be rubbish. That's why the data and the model must be good or perfect, that you can expect you have a good data. This is the data that you will have, and when we convert it, we will convert it to the Excel. The Excel can be read by the SIMCA or the Metabo analyst. This is the software that we do the multivariate data analysis. For this, the multivariate data analysis, it will, because every one spectra, it will be as one dot in the score plot. It will reduce the dimension. As we see here, all the sample, the one that tend to be similar, they will be, let's say you have five replicates from one group, then you can expect that they will be similar. They are sharing similarity and they are different from other group. That means they will cluster together as different from other group. The group they are clustered together and they have distance from other group. They will be sharing differences. They have differences from other group. Just now I mentioned to you that for the multivariate data analysis, we have unsupervised analysis, which is blind. We don't mention to the system anything about our data. We just ask the system, which is the PCA, mostly the PCA or the HTA, to cluster our data based on the metabolite. What are the metabolite? The one they are differences. The one they tend to be similar, they will cluster together. The one they are different, they will cluster away from the first group. And for supervised method, this one, when we have other data, the one we don't analyze it from our system, we move to correlate using the PLS, or we mentioned to the system that we have different group. We told the system that we have group A, group B, and group C. The system already know there is some differences from our data. Then the system try to separate the data based on the knowledge that we provided to the system. That's why it is supervised. Then this supervised method, we need to do validation as different from unsupervised method, which is no need to do any validation. This is the score plot, which is the map of observation. In the score plot, for the multivariate data analysis, we see if we have 3 groups, every group has 5 replicate, then we will see 15 dots in the score plot. That's when all the sample, they will be in the score plot. Every one spectra has 1 dot. And then the differences we can see that the blue, they are separated from other. The one they are all clustered together, they are sharing some similarity, and they are different from the blue, the group with the blue color. And the loading column plot, this is to show that the metabolite, the one into the contributing to the differences between the groups. And this is based on the position, because the loading and the score plot, they are complementary to each other. In the left quadrant here, this is the metabolite, the one contributing to the blue, to the separation of the blue group from other groups. And then vice versa, all the one they are on the right, they are contributing to the separation of other group from the blue color. That's why the loading and the score plot, they are complementary to each other. We need to present both of them. Then we need to discuss both of them as from the score or the loading plot. There is the concept of the scaling. When we have, let's see, we have 3 different signals. The one they are negligible, which is like the noise. Sometimes when we compare between different groups, and we see that the small peak, the one they are contributing to the separation between the groups, and we don't want to lose the information coming from that signal, then we need to introduce the scaling method. We need to force the information coming from the small peak to be important, or to be as important as the major peak. That's why we need to do the UV or the Pareto scaling method. I think I don't have time to discuss more about this, but if you need more information, I can provide it to you, or you can ask question later on. This one, when we have the PCA from the PCA, and we have 3 different groups. As I mentioned in the beginning, when we have different batches, let's see here, on the right, the B, they are the same group, but we have the sample from different supplier or 2 different batch. You can see they are totally separated by PC2. We have the first batch is separated from the other batch of B, or C and A, they are separated from A and C. This is when we have 2 different batches, we need to be careful with this. That's why I just show this example to show you when you have totally different batch, it can cause to have differences within the group. And in that case, maybe you can try to have different combination of PC1 or PC2, because this is 2 dimension. You can have the combination of PC1 and PC2 that show you that they have 2 batches. But if you want to avoid that problem, you can have the combination of PC1 against PC3. Then that information, it will not appear. For the scaling method, we can see that, and also for the loading plot, we can have the loading column plot, that which are the compound, the one they are contributing to the one on the right or on the left. The one on the top, or which is in the positive side, they are contributing to the separation of B from A and C, and the one on the bottom, they are contributing to the separation of A and C from B. I think this one I just want to show what is the difference when you have 3 different scaling method. It can give you totally different result. And also from when we don't have any scaling method, you can see that there is no separation between the batches. But in this case, we don't have that much differences, but in some result or in some data, you can have totally different result. That's why sometimes you can have complementary result from different scaling method. Maybe you can get some different information, and you can discuss it separately. Before we introduce our result to the multivariate data analysis, we need to go for PCA. the first thing we need to go for PCA. If you don't have no separation, then you can proceed with the PLSDA. But for publication, don't go for PLSDA without presenting the PCA, because they will ask you why you don't go for PCA as unsupervised method, why you go for supervised method. If you have a good separation with the unsupervised method as a PCA or SCA, then it's better to just proceed with the PCA. But for the PLSDA, if you want to have the VIP, which is the variable important in the projection, can be done only with the PLSDA or OPLSDA. If you need further information, you can ask me later on, or you can contact me. I have some cases, some case study from my previous publication for me and for my student. This is one we combine between the MS data and with the NMR data. You can see they're sharing the same clustering. We can, when we are comparing between the different ratio, the water extract, the 50%, and the ethanol extract, we can see that most of the MS, LCMS, and the NMR showing the same cluster. That's 0% is separated for 0 and 50 is separated from 100, which is on the left. In NMR is the same thing, that we have clustering, 3 different cluster. And from the MS, you can see there is many metabolite, but the one we identify is less than the NMR. But in most of the NMR publication, if you don't run the LCMS, they will ask you, you need to at least you go for profiling for the LCMS to validate the identification of the metabolite that you identify with the NMR. That's the better way is to do it complementary to each other. And this is for the PLS, when we correlate the data based on with the bioactivity. When you have the bioactivity here, we have the DBPH, which is the antioxidant activity, or alpha glucosidase. Then we want to see the bioactive compound, the one they are contributing to this, to the separation, and also to contributing to the bioactivity. We can see, because we know that the water extract has highest activity in this case, and this extract has the highest alpha glucosidase activity. We can see it is a normal correlation. We can see that the compound, the one they are higher in the water extract, we can suggest them as the bioactive compound or the chemical marker for this plant. Another example, I compare between different variety from different location. This one, ficus, for my PhD student. We compare between different variety. We know which variety give the best activity, and also which one give the most metabolite as compared to the other, and which location give the significant metabolite as compared to other location. We find that from the profiling, it's very difficult for us to see which are the metabolite. Even you can see some metabolite, there are differences based on the profile. But if you want to see the overall or most of the metabolite, is better to do the multivariate data analysis. From the multivariate data analysis, we can see that the group, the one they are clustering together, and FDA, which is ficus deltoidea, and the one from Terengganu, the one they are separated from other varieties, and the one they have the best activity. Another optimization can be done with the metabolomics, which is I have different growth stages, the plant from different growth stages. I have the flowering stage, before and after the flowering stage, and have phyllanthus with different species, different drying methods, and different solvent extract. This is from my publication. You can see that when we compare between the different growth stages, we have 8 weeks, 10 weeks, and 12 weeks. 10 weeks is the flowering stage, because from most of publication, they mentioned that the flowering stage is the stage where the plant give the best activity and give most of the phenolic compound because the insect can attack the plant during that stage, and because the plant can produce those metabolites to avoid the attack of the insect. And we can see that from our result, you can see that 10 weeks, it is correlated with the bioactivity, and also all the metabolites, especially the phenolics, are correlated with this bioactivity, and they are considered as the biomarkers, mostly the phenolics. When we compare the different drying methods, then we can see that the freeze drying, the one can preserve most of the metabolite in the plant, and this freeze drying has most of the phenolic compound as compared to the oven drying and the air drying. And we can see from the loading column plot, the compound, when they are higher in the freeze drying as compared to the oven drying and the air drying. We have the same extract when we optimize it. We try to take the flowering stage at 10 weeks, and also we extract it with the solvent ethanol 80%, and also we dry it with the freeze drying, and we introduce it to the treatment for the obese diabetic rats. We quantify, because most of the publication now, they ask you to standardize the extract. You need to see all the compound that you find them as the biomarkers and try to do the absolute quantification. You need to know the amount of the extract of the metabolite in your extract. We see in the doses that we give to the rats, 250 milligram per kg and 500 milligram, and we see what are the compound and the amount. This is generated from my publication. When we compare between the normal diet group and the high-fat diet group. This high-fat diet group, for 3 months, we feed them with the high-fat diet, and then we induce the diabetic with STZ, and we treated them for 4 weeks using the Philanthus, the one I showed you just now, and we compare with the positive controls as the metformin. This is just to show the weight when they become obese. After 12 weeks, they become obese, and they are significantly different between the obese group and the non-diet group, normal diet group. And the biochemical, I just show some compound, the one we do, glucose. We see that the high dose give the highest effect as compared to the low dose, and also the metformin. We can see that they are significantly different. It can lower the LDL and increase the HDL, and lower the cholesterol, triglyceride, and lower the creatinine and also the glucose. When we compare between the trajectory between before injection of the STZ and the baseline after we inject the STZ when they become diabetic, and after the treatment, we can see that the shifting from here. This is the diabetic group before. This is before diabetic, and the one on the right, they are diabetic, and after the treatment of 4 weeks, we can see they are shifting towards the normal rats. We can see the same trend of the metformin on the left side. Here, when we have 2 different doses, we have the low dose and the high dose of 500. You can see that the 500 milligram per kg can give effect, and it causes shifting to the treated rats towards the normal rats. It didn't give the same mechanism, but at least it's going away from the diabetic group as compared to the low dose. The low dose is shifting from the diabetic group, but it doesn't have the same effect or the significant effect as the high dose. This is the metabolite, the one they are contributing based on the VIP value, and also the one they are significant. We can see that the taurine, the choline, and the glucose, creatinine, all these metabolites are the biomarkers of diabetic and also for the treatment of this plant. Based on the significant metabolite, we come out with the mechanism of, we see which pathway they are involved with the significant metabolite. And from the metaboanalysis, we can see what are the metabolites when they are significant. And we discuss the metabolite with the highest impact. We can see that the synthesis and degradation of the ketone bodies, they are the most impactful pathway, and we discuss it based on that and based on which metabolites are involved and they are significant in this pathway. And then we try to come out with our design of the pathway, because we cannot take that pathway because it is the same as previous publication. We need to design our own pathway. That's why we need to come out with different design as compared to other publication. Maybe you can go for different publications, see how they design it, and you can design your own. See the biomarkers, the one they are contributing to the pathway, and also the contributing to the treatment. This is my current study, what I am doing now. All the plant that I'm studying, especially focusing on the obese diabetic, and also sometimes I'm dealing with the inflammation, and also for the cancer study, using mostly the metabolomic study for the NMR and also for the LCMS, and sometimes for GCMS. For the concluding remark, I can give some points that the metabolomics is a tool, but not objective. When you are designing your experiment, you need to design it properly, and you need to answer your questions, because the metabolomics can study what are the differences between your sample based on the metabolite. And when you design your experiment, you need to have clear and very critical design, starting from the sample preparation until the pathway analysis. You need to have very clear and very specific study, especially when you are dealing with targeted or non-targeted. If your study needs to go for untargeted, then you cannot go for targeted analysis. And you cannot do the metabolomics without collaboration. You need to collaborate, because it's dealing with chemistry, pharmacy, biochemistry, and also pharmaceutical study. That's why you need to collaborate, and you are okay to collaborate with us or with any people doing metabolomics. And also you can do complementary study as compared to the proteomics, genomics, and you can correlate between the studies, and you can have more information. With that, I want to acknowledge my group from Symbiosis and from UPM, and my ex-supervisor, Prof. Farid Abbas from UPM, and my group from UPM. When we have fun, always fun. Now they are busy with the convocation, and also here in UPM we have convocation. They are busy. That's why today they cannot join. Sorry for that. With that, we have a service for all the omics approach. You are welcome to send your sample, or you come for workshop or for collaboration. And we have upcoming RCSB conference in Malacca from Johor. It is not very far. Please join us in Malacca on 20 to 21 December this year in Swiss Garden Hotel. You are welcome, and I hope to see you there. Thank you very much. Any questions? I will be happy to answer them. Thank you very much.
Okay, thank you very much, Dr. Ahmed Mediani, for a very interesting topic on metabolomics, focusing on natural products. So, okay, now I proceed to the questions and answer sessions. So, I think, can I have one question from audience? Okay, I'm reading the questions from Maizatul Akmal Yahyu. So, thank you, Dr. Ahmed Mediani, for your sharing. I have a questions here for metabolomic annotations. So there are numbers of library or database available like MassBank, Medtrack, and so on. So do you think those libraries give the right compounds?
Okay, thank you very much for the interesting question. Yes, especially for the MS, this database, they are for the MS library. They have some, but mostly they are for the biofluid or for the animal study. If you are going for the plant, it is not rich. And also for the NMR, we have the same problem. when we are dealing with the plant sample, we have a problem when we want to identify the compound. You need to go for the literature review. And also you need to go for some library like the Genomics. They are rich with the biofluid sample, metabolite, but they don't have any compound coming from the secondary metabolite of the plant. If you are dealing with the primary metabolite, it's very easy for you to identify. But if you are dealing with the secondary metabolite, it's very difficult for you. And to be honest, if you are doing metabolomics on the plant and there is no study done on that plant, it's very difficult for you to do it. It's better to go for the plant when they do some isolation, or at least they have some dereplication study using the MS or the NMR. Okay.
Yes, Dr. I totally agree with you, because as far as I know, so far actually dealing with this secondary metabolites for annotation, especially for the plants. So I think for me it's a big challenge for me because there's time consuming to annotate the compound from plant, especially as you mentioned, very rare plants. They have not many publication about that plants. Okay, so now we move to the next question, please. It's from, I think, from Ida Madiha. Salam, doctor. Thank you so much for your sharing. So we would like to ask how robust the results from the control condition at Mr. Laboratory compared to the actual processing in industrial scale, since the plant is very sensitive, and how it's collected, handled, and processed. Thank you.
Okay, very nice question. Thank you for the very interesting question. Yes, that's why for the industry also, they need to control most of the factor that can affect the metabolite. When you are dealing in the lab, you are doing with the small scale. But when you go for the industry, they need to deal with the same supplier, the one they can trust, or at least maybe they can have a farm. They can plant their own sample. Then at least they can handle, or they can control most of the factors they can affect. Or when they receive the batch, they need to have the method or the model that they need to test that batch when they come to the industry before they can proceed with the drug. They need to test it or analyze it before they can accept or reject that sample from the supplier. You just need maybe to profile and put in the model. Then you see that if the sample is clustered with the same sample from the previous batch, then they can accept the sample. If it is not clustered the same thing, or they don't have the same profile, then they can reject or go for the different supplier. Or they can plant their own sample is the best way. The best solution is to plant your own sample and you control most of the factors. Then at least you will be confident. I hope I answered your question.
I think, yes, I think Ida Madiha. So probably we can go to the next question, please. So it's from the Suryani Shahid. So what do you think of the pharmaceutical industry and research directions? Are we at a par at least neighboring countries?
Thank you for the question. I think pharmacy, now they start to introduce, or they use these approaches like the metabolomics or the omics. They are at least now they are in line with other companies for overseas. At least last time when I start, to be honest, when I start my PhD on 2010, most of the pharmacy people, even the one they are from the same university, they are against this approach. They don't like the natural product people. They are against this omics. They say they don't trust this metabolomics or this omics approach. They want to go for the isolation or the reductionist approach before. That's why, but now at least they are happy with this approach that they are using. And most of the pharmacy or the industry nowadays, they are using, and they are collaborating with the university. I think nowadays Malaysia, it is in line with other overseas companies.
Yes, I totally agree with you, because actually at the moment, if we are looking for the for new compound or bacterial compound. So the best first we have to do is metabolomy, right?
Yes, to do the dereplication. At least no need to waste your time. Maybe sometimes if you are dealing with the same compound, why you want to waste your time? If you do the dereplication using the metabolomics, you know that you are dealing with the new compound or the previous compound, the one already reported before. Then no need to waste your time and the cost, saving the cost.
Yeah, I think if you non-compound, better you buy the new compound and test it. That's it, right?
Yes.
No need
exactly.
to waste. All right. Okay. Next question, please. I think we have some question. Okay. Assalamualaikum, doctor. Good evening. So I think it's from Muhammad Amirul. So what the difference between primary and secondary metabolites? Thank you.
Okay. Thank you for the question. When we are comparing between the primary and the secondary metabolite, for the human, we have only the primary, or the animal, we have only the primary metabolite, the one they are dealing with the reproduction. But when we deal with the secondary metabolite, the one we can take it from natural product. This compound can be found only in the natural products or on the plant. When the plant can produce this secondary metabolite to avoid the insect or the attack of the insect. As example, for the primary sugar, the amino acid, the fatty acid, which is found in human or animals, but the secondary metabolite like the phenolics, the flavonoids, or other compound, the one the plant are producing. I hope I answer your question.
I think yes. I think here the last question, please. Okay, good afternoon, Dr. Ahmed. Thank you so much for sharing. So based on your experience, what are the most critical parts in the metabolomic study? Thank you.
Okay, thank you for the interesting question. For me, the metabolomics, all the steps are very important, but the most important step is the design. When you design your experiment very nicely and very critically, then you will not have any problem during the latest part or the latest section. There is so many students or many researchers when they don't design properly the experiment, but when they go for when they reach the last stage, when they do maybe they focus on the targeted analysis, but when they do the extraction or the processing, when they go for the metabolite identification, they found that they don't identify the compound of interest. They need to go back and do the untargeted, or they need to change totally the extraction method or the processing method. That's why the design is very important. And also, when you have to do the multivariate data analysis, you need to do the statistical analysis, and also you need to deal with the biological replicate. Sometimes the people, they are not doing the biological replicate, they're doing only the technical replicate. When they go for the biological, for the multivariate data analysis, when they do the multivariate data analysis, they will see all the sample, they are clustering exactly the same. They are only like 1 point. They cannot publish this result because they don't do any biological replicate. Especially for when you are dealing with the anima. I have one student last time. He just processed the same sample. He processed it different time, and he considered them as 6 biological replicate. When I see his result, I put it in the SIMCA to do the multivariate data analysis. I see that all the dots are clustered all together as 1 point. We don't see any replication. Then I ask him to start over from the beginning. It's like wasting time. That's why the design is very important in the metabolomic study. I hope I answered your question.
Yeah, I think thank you for your question and answering. So I think we have finished with the questions and answer sessions. And thank you very much again, Dr. Ahmed from Inbiosis UKM for very nice sharings and informative informations, especially regarding the metabolomic as holistic approach in natural products. So we hope we can make a scientific collaboration in the future between IBD and also UKM,
of course,
inshallah.
Inshallah.
I think that's all from me. So again, thank you very much for your time and so on.
Thank you very much. I really enjoy. Thanks. I'm grateful for this
Thank you,
invitation.
Ahmed. So
Thanks.
maybe we can make a collaboration in the future.
Okay.
All right.
Bye.
Okay.
Thank you very much. Okay.
Assalamualaikum.
Alaikum salam wa rahmatullah.